people ask about vasectomy and want to learn more, but we also are always thinking about what are the alternatives? What other options are there that we know that a vasectomy may be the best choice for a family who's complete or a man whose family is complete, whatever that number is. But there's a long time of fertility when it would be wonderful to have other options. And there's been a lot of talk about other options. There's been discussions, there's been a lot of starts and stops, and nothing has entered the marketplace. I think condoms have been around for thousands of years. I think vasectomy has been around for 125 years. So trying to find a new product for the marketplace for men before their family is complete is something everybody, I think, is interested in. So I'm very honored to introduce our next speaker. His name is Kevin Eisenfrats. He's the co-founder and the CEO of Contraline. It's a medical device company and developing world's, what they hopes to be the first in the market, long-lasting, reversible male contraceptive using advances in the hydrogel technology. I won't go into more detail with time is short, but I introduced Kevin, and Kevin is going to take us through for the next half hour. There'll be time for questions and answers as well. So Kevin, please take the mic and start sharing what your program's all about.
Awesome. Thanks, everyone. Thanks, World Vasectomy Day team. And it's great to meet everyone here. Really excited to talk to you for the next 45 minutes or so about the future of male contraception. Obviously, we don't have a product on the market right now, so a lot of this is the advancements we're making in the field, and hopefully get you all excited and maybe even involved in helping us advance the whole field. So a little bit about my background. I started this company in 2015 specifically to work on advancing male contraceptives. I started the company with a professor named Dr. John Hare, who's also the creator of the Sperm Check vasectomy at-home sperm test kit. So if those of you who are familiar with the at-home sperm testing, that came out of the same lab that this company came out of. And we've made a lot of advancements from concept into now, early next year, we're actually starting the first clinical trial on our product. And it's going to be the first clinical trial on a new male contraceptive device in over 20 years. So there's a lot of exciting things happening, and I'm going to share with you more about what we're working on at Contraline. Does everyone see my screen okay? Okay, so starting off with just some disclosures, I am the co-founder and CEO of this company, and I'm also an inventor on quite a few patents relating to male and female contraception. So I think it's important we start off and talk about the global problem that we're trying to solve, whether we're talking about vasectomy or we're talking about the future of male contraception. We're all working on the same team to reduce the number of unintended pregnancies worldwide. So right now, 64% of pregnancies globally are unintended, which equates to about 121 million pregnancies each year. In the United States, it's about 45% of pregnancies are unintended. So clearly, even though there's dozens of female contraceptives on the market, even though 500,000 guys are getting a vasectomy in the United States each year, still the rate of unintended pregnancy is very high, almost half of all pregnancies. And in the United States, 1 in 4 men who have had intercourse has led to an unintended pregnancy. So this is still a huge issue that's not solved. And I think the problem is the fact that men just don't have enough options. There's 2 options right now available, as we all know. There's condoms and vasectomy. Condoms have a high dissatisfaction rate and a high failure rate of about 13% globally. And vasectomy is still considered a permanent procedure. So therefore, it is really being used by men who are family complete. As Jonathan was talking about earlier, if you're a man who is not family complete and you don't like using condoms, really, you don't have many options. You're either relying on the female partner or you're relying on even the pullout method, which we all know as kind of the experts here that's not a great method to be solely reliant on. So some statistics to share with you. This is a study in the United States that the CDC published. They look at contraceptive use over time amongst couples. And this is a study specifically in single men. What you see is that about 82% of single men are relying on contraception, which is great. And in fact, among single men, 60% of them are using male-based methods as opposed to female methods. But when you start to look at the breakdown of those methods, you look at condoms from 2002 to 2015, condom usage has pretty much stagnated, and vasectomy usage has pretty much stagnated. In fact, there's a couple of papers more recently published by Mike Eisenberg out of Stanford, for example, who are talking about how the vasectomy rate in the United States is even decreasing. But the crazy thing is that the pullout method or withdrawal has actually almost doubled in the past 2 decades. And so we're looking at about going from 10% to almost 20% of single guys are now relying on the pullout method in the United States, which is highlighting to you that how important it is that we actually have effective male contraceptives, and the fact that men want to play a bigger role, but they can't because there's not other options available to them. So now I want to talk a little bit about what's been tried before. We're going to talk a lot about this concept of vas occlusion or vas occlusive contraception. The concept of vas occlusive contraception is simple. What happens when you put a material, a gel, or a different type of device inside of the vas deferens to block sperm? So basically, similar mechanism of action to a vasectomy, but we're not completely or permanently occluding or severing the vas deferens. Instead, we're putting a device inside. And this concept is not new. In fact, I believe it was the late 1970s that there started to be papers published on vas occlusive contraception. But you can see at the bottom, I listed a bunch of citations here on what's been attempted before. There's probably been maybe 5 to 7 different methods that have been attempted at vas occlusive contraception over the past 50 years. And so my company Contraline by no means is the first company to work on this problem. We just have a new way of solving this problem. So let's talk a little bit about what's been done before. Well, I believe it was in the 1980s or 70s. In China, they actually tried to inject polyurethane in the vas deferens. And it was actually very successful. They injected 12,000 men with polyurethane, and they showed that it had 98% azoospermia rate. But the problem was that polyurethane actually is a carcinogen. It causes cancer. And so they found it to be very toxic, and they discontinued that product indefinitely. But what that showed was that there's proof of concept of efficacy that you can inject a material in the vas deferens and cause azoospermia, just like a vasectomy. There's been attempts at looking at injectable silicone rubber. So we know in the medical device industry, silicone is a much safer, non-toxic material. It's used in other devices. But the problem with silicone is that it wasn't very effective at blocking sperm because the vas deferens is a peristaltic pump. It basically pumps sperm, and the silicone was a very hard material that wasn't elastic. It wasn't able to expand with the vas deferens, and so sperm would just travel around the silicone rubber. It also was very difficult to make this rubber material, and there were other complications with it, and so their efficacy rate was very low, and that research was stopped. And that was done both in Indonesia as well as the Netherlands. There's been attempts at using implantable silicone plugs where these silicone plugs are actually preformed, they're not injectable. And then you can actually kind of put them through a needle into the vas deferens, or maybe even make an incision in the vas deferens and then put the plugs in and then suture those plugs to the vas deferens. This is a very invasive procedure. It's actually took a lot longer than a vasectomy would. So injectable is definitely much more preferred over implantable. But also sperm would travel around the silicone plugs as well. So silicone has just not been shown to be a very good material in terms of efficacy. And then some of you probably are familiar. I mean, there's been about 2 decades now worth of research on products called Rysug in India and Vasoljel in the United States. And both of these products use a polymer called SMA. It stands for styrene maleic anhydride, or styrene maleic acid, which is a slight modification of it. And what these polymers are, they're basically like plastics. And so when you dissolve SMA in a solvent called DMSO, this is an organic solvent, you inject that in the vas deferens, the SMA actually wants to precipitate out of the DMSO and basically form this plug inside the vas deferens. Now, this research has been going on for decades, and they've shown to have really good proof of concept, but there's been quite a few challenges. The first is that this SMA polymer or plastic, it's pretty difficult to reverse. And so there hasn't been great data to show the reversibility. They found that the SMA has a pretty toxic effect on sperm and is actually affecting the acrosome of the sperm. So even when you are able to remove the plug out of the vas deferens, they have seen some side effects, or some, should I say, effect on the sperm itself. So the forward progression and the motility of the sperm does not restore to normal levels. And that's something that needs to be explored more in the future. So more work is being done on this SMA polymer, but the big difference is that it's in an organic solvent that they're injecting this in. So if you look at kind of from a medical device approach, right, if all of you were to develop a new vas-occlusive male contraceptive, kind of what are the lessons learned? Well, I think the first thing is that we need to have a reliable entry in the vas lumen. No matter how you're implanting this material, whether it's injectable or implantable, the doctor needs to be able to get inside the lumen reliably and know they're inside the lumen. We also need a material that's easily injectable. One of the challenges with the SMA polymer, for example, is that it's extremely viscous. And when you have high viscosity, that can pose challenges like difficult injection or difficult formation of the gel, or you don't know if you're injecting enough or too much, or the gel actually is leaking into other areas like the fascia or in the smooth muscle where you don't want it to be entering. Obviously, for this to be effective, it needs to block sperm effectively. The gel has to be durable, but also elastic. It has to be able to withstand the pressures inside the vas deferens, which is something that silicone was not able to show. It's got to be safe. It has to be biocompatible and non-toxic. Something that we've really innovated on at Contraline is the ability to do consistent injection volume and plugs. So when you have different patients, you don't want one patient to have a gel that's only a couple centimeters and another one to have a gel that's a lot bigger than that. You want a reliable device within each patient. And so that the device is going to be safe and effective and reversible. Now, when we talk about reversibility, the reversal has to be a minor procedure or minimally invasive, and also has to be safe. We can't be injecting, a non-biocompatible chemical into the vas deferens to dissolve this gel. It has to be something much safer. And then once you reverse it, it's not only important that the sperm come back to normal concentrations and sperm counts, but we also have to be looking at the sperm motility and viability. So do we have long-term restoration of fertility, right? And so this is kind of the design criteria, if you will, on creating a new male contraceptive product. So let's talk a little bit about Contraline and our product, what we're developing. We're working on a product called Atom. That's the name of our product. It doesn't stand for anything. It's just the name. And it's what we're trying to achieve is a long-lasting, non-hormonal and non-permanent vas-occlusive contraception that uses a hydrogel to occlude the vas deferens. Now, a hydrogel, as far as we know, we're the first approach at solving this problem using a hydrogel approach. So let's talk about what is a hydrogel and why is that so innovative and such a better approach. So a hydrogel is a water-soluble, easily injectable, durable, and highly biocompatible material. Our gel that we're implanting is actually about 90% water and 10% this polymer network or matrix. And so when you talk about safety, it's extremely safe. In fact, there's a number of medical devices that are on the market that use hydrogels, but they're using them for completely different applications. And so we're the first team to ever think about using a gel specifically and making it for the vas deferens. Our gel is also easily injectable. It's about the same viscosity of water, so you're not facing challenges getting this stuff into the vas deferens. Once it's implanted, it's already a gel. It doesn't take hours or even minutes. It actually takes seconds to be implanted and form that gel. And it's extremely durable. It's not going to migrate around. It's not going to go anywhere. It's actually able to withstand the forces and the pressures of the vas deferens. Once it's implanted, this gel has very small pores. We're talking about the size of nanometers. So it's able to block sperm from traveling through. And one of the biggest innovations behind this gel that what we've created is that it's actually temporary. So what I mean by that is that this gel is able to degrade over a long period of time. It actually lasts for a long period of time and at some point degrades. And so what we're talking about is almost like a temporary vasectomy, if you will, something where you have effective occlusion, but then after a certain number of years, it degrades and the patient's fertility is restored. And what's really cool is that we can actually tune that lifespan. So we have different chemistries of that gel that allow us to achieve, if we wanted to do a 6-month vasectomy, for example, or a 1-year long or a 2-year long. And hopefully one day we're going to do a much longer amount, 3, 5+ years in terms of the lifespan of this gel. We're actually starting, our first product is going to be a 1-year long male contraceptive. We're also optimizing a procedure that we can remove this gel on demand through a minimally invasive procedure. So we can remove the occlusion before that end of the lifespan if the patient desires. So from a very high level, from a just purely conceptual, this is what we're working on. So then we wanted to take it one step forward, which is now that we have this gel, how do we create a procedure for the physician and basically make this entire procedure reliable and consistent and easy, that anyone can do this procedure. So what we've created is the first delivery device for Atom, for our hydrogel. And so what you have here is a really simple, reusable and rechargeable device that can be used anywhere in the world, where basically we have these 2 syringes that clip in and that form the gel. The gel is formed by these 2 polymers that cross-link with each other. And what you'll see on the next slide is as simple as cannulating the vas deferens with an over-the-needle catheter. And then using this, this device, we can basically lure lock this end of the device into the cannula. And this device will then inject the same amount of gel into every single patient every time. So we've basically created an automated device that's consistent and simple. And so there's a consumable portion, and then there's a reusable portion to the procedure as well. So I have a quick video to show. It's on a model, on a no scalpel vasectomy model, where we're going to be showing how we actually perform our procedure. The important thing to note is that our procedure initially is pretty much identical to a no scalpel vasectomy. And so the only difference between our procedure for implanting Atom and the no scalpel vasectomy is that once the vas is externalized, the physician is simply inserting an angiocatheter or an over the needle catheter. We're using a 24 gauge angiocath to cannulate the lumen of the vas. And then it's as simple as hooking up this delivery device. So I'm going to play this video here. Again, this is a model, a mannequin, and also this is not a physician performing this procedure right now. So excuse if there's any kind of inconsistencies with the no scalpel vasectomy. Once the angiocatheter is in the lumen, the physician's actually able to kind of pull the catheter in and out gently, and they know if they don't feel resistance, then they're in the lumen. Right here, you can see that the user is just taking off a cap. The device is already primed. There's no dead volume inside or anything like that. They can hook it up, and then they press in the trigger on the front, and then the injection is performed. And that injection process takes about 5 seconds. In our clinical trial, we're actually holding for a little bit longer than that just for safety measures, but overall, this entire process is very quick. So now that we've talked from a very high level how this procedure happens, I want to share just a little bit of data with you all about our device and the performance of it. So we have done a lot of studies. Please excuse this dot over here. That's not part of my graph, but we've done a lot of testing on this device to show that it's safe and it works. And in our studies, canines or dogs are the best model for a couple of reasons. The first is that the procedure is very similar in dogs to humans. In fact, it's actually a little bit more invasive because the canine scrotum is so thick, we're actually not able to do a no scalpel vasectomy on the canine model. So we've had to do more traditional approaches and make larger incisions to access the vas deferens. Secondary, the vas is actually pretty deep within the spermatic cord compared to humans as well. So in fact, I would argue that canines have been a little bit more of an invasive model than what we expect humans to be. And then lastly, dogs have a lot more sperm than humans. So if you look at this dog as an example, what we're looking at is the baseline, which is the pre-zero time point. The sperm concentration is on the order of 1.5 billion sperm per milliliter, which is about a couple orders of magnitude higher sperm count than humans as well. And what we've seen is that when we implant ADAM into the vas deferens of these dogs, we actually are able to see the sperm count drop down to 0, and we have azoospermia as soon as 14 days after implantation. And just so you know, 14 days was actually the first time point we tested. So potentially could be sooner, but what we're looking at is azoospermia around the same time frame as a vasectomy. And what we've seen is that the azoospermia is continuous. We do sperm testing pretty much twice a month. And we have dogs who have been going out now for 2 years. This is one example of a dog who's, I think, at 1 year where we can see azoospermia and also 0% motility. We've done a lot of safety testing. As this is a medical device that's going to be implanted into humans and last for a long period of time, we want to know that there's nothing that gives us concern. One of the beauties of hydrogels is the fact that they're so safe. So we've done a wide variety of safety testing, from cytotoxicity, irritation, sensitization, systemic toxicity, pyrogenicity. We've implanted this into dogs and looked for years at the histopathology. And we've also even done reproductive mutation, where we worked with Larry Lipschultz at Baylor, and we looked at the sperm that interact with their gel. Are we causing any chromosomal mutations? And we used FISH testing to do that work. And what we've seen across the board is that we have had no issues for biocompatibility for both our gel, the injector, and the degradation products of the hydrogel as well. So over time, we're not seeing any safety issues. So with that, I want to shift gears a little bit and talk about how do we get ADAM to market? You know, people have been trying to work on this problem for decades now, but how do we as Contraline actually bring this device to doctors and to patients? So the first thing I'll talk about is that we are going to we're going to clinical trials early next year. We're anticipating starting our clinical trials in March of 2022. And like I mentioned earlier, this is going to be the first clinical trial on a male contraceptive device in over 20 years. This study will be done in Melbourne, Australia, and we have 2 sites and 3 physicians who are going to be performing the procedure there. And we'll be implanting ADAM into up to 25 men and following them for 3 years. And now the reason for 3 years is because we want to test the safety and efficacy of this product for a long duration of time. We want to start knowing how long does this gel last inside the body. Primarily, this is a safety study. Again, this is proof of concept here. So we're really looking at the safety, adverse events, things like granulomas, infection, hematomas, pain. So we're looking at all the safety endpoints, but we will also be looking at the feasibility of the implantation. How successful is the doctor at getting the gel in the vas deferens? And we'll also be monitoring the semen parameters. So how does the sperm count, the sperm motility, viability, and volume change over time in these patients? Obviously, we also will be looking at, we're only recruiting men, we're not recruiting couples. However, if that male patient in the study has a partner, we'll also be looking at pregnancy outcomes as well. So once we have some data from this first in human study, we're not going to wait the full 3 years before moving on to the next phase. We are going to be starting a pivotal clinical trial that will support our FDA approval, and that pivotal trial will likely start in 2023 in the United States, and we'll also likely have some international sites as well. Ultimately, we hope to get FDA approval on our product in the United States by 2024, maybe 2025 at the latest. And then we'll have commercialized the technology and start international expansion to other countries. Obviously, United States is not the only country in the world that wants this product. We know that for a fact. However, we do feel that if we are able to successfully get FDA approval in the United States, then it'll be much easier and other countries will follow, because this is the FDA in the United States has such a high barrier to entry. And so we've worked with the FDA closely for years up till now, and we plan on continuing working with them closely over the coming years to make sure this product comes to market and it's safe and effective. I want to talk about the demand for new male contraceptives. There's a lot of market research that we've done, as well as that's published on the demand out there. In a survey that we've done, this is not published data. We've done a survey of 1,005 men in the United States, ages 18 to 65. And we have found that 52.2% of men were very or somewhat interested in receiving Adam. And their most desired lifespan of the product, how long they want the gel to last, is about 1 to 2 years. And so there's a lot of demand here, but also clearly men want something that is a little bit more temporary than a vasectomy product. And then in a survey in 2005, now this is a survey that's very often cited in our industry. This was a survey done of 9,000 men across 9 different countries down here at the bottom. And they found that 55% of men would be very interested, but they were asking about different forms of male contraception. They asked about implants, injection, jellies, and pills, a male pill. And so the data actually from that study in 2005 looks very similar to ours. And the most desired frequency was about once a year, meaning that men really wanted a product that could be administered once a year. And then here's what's amazing. Contraline, we have received over 1,700 signups from men to be in our clinical trials to date, and we have done zero marketing. We have not posted on Facebook. We've not posted a single tweet or TikTok. We have had over 1,700 men from around the world ask us if they could be in our clinical trials. And the highest demand from men comes from men ages 25 to 34 years old. So these are men right now who are not really considering or actually serious about getting a vasectomy. And so that is the age group that we feel has the most need, has the highest unmet need for our product. There's a huge impact that a product like Adam could have. There was a study in 2018 that Emily Dorman published, where basically she looked at 3 different countries, the United States, South Africa, and Nigeria, and she assumed that if only 10% of men who are interested in receiving a vas occlusive actually went and got it, so that's pretty conservative, then this would decrease unintended pregnancies by 5.2% in the United States, 5% in South Africa, and 38% in Nigeria. And we're talking about hundreds of thousands of pregnancies in the United States, millions globally. So there's a huge global impact that our product can have. So my last slide to close out the presentation, and then I'll open up for Q&A, is there's a lot of physicians on the call right now. There's a lot of vasectomy performing doctors here. We're not on the market yet. I'm not here to sell you on our product, Adam, and why you should be performing it. However, if anything I said today might have interested you, or you want to get involved in helping us get this product to the market, there's a couple of different ways that you can help. The first one is that if you visit contraline.com/participate, we have a program called a letter of interest, or letter of intent, and it's non-binding, it's non-legal, and basically you can quickly fill out this form. It takes about 10 seconds, and tell us who you are, where you're based, and it'll basically say, once we're on the market, we will reach out to you about getting Adam into your clinic or your hospital, and you can be involved in being one of the first doctors to do our procedure. Now, the second point is, if you're a clinic or a center or hospital that does clinical research and you love doing clinical research, we're seeking potential clinical sites for our pivotal trial next year. So if you want to be involved in the clinical trials for this product, we'd love to talk to you. Third, if you'd like to consult, advise, or be involved in our product development, maybe you're entrepreneurial or inventive, we'd love to talk more with you and innovate with you. So my email is kevin@contraline.com. You're more than welcome to reach out to us. We work with physicians all over the world actively, and so we're always looking to get more involved with others. And then fourth, please, if you're interested in what we're doing, advocate amongst other physicians and patients the broader need for new male contraceptive options. I think this is a team-wide approach, and all of us are here on the same page to reduce unintended pregnancies. And so I think that all of us need to be cheering for and helping each other kind of get these across the finish line. So thank you all so much for your time here. And I'll now take some questions and hopefully try my best to answer them.
Kevin, thank you very much. Jonathan wanted me to moderate the questions a little bit. You can see them in the chat if you're hitting that. Some of the questions coming in, rather than me picking them, are any of them particularly engaging for you? How can we invest in Contraline? I'm interested. We're all interested, I suppose.
Yeah, so I'll start going through the questions here and see if any of them stand out. So someone was asking how many pregnancies end in abortion in the United States. In the UK, we think pro-life movement of anti-abortion is very strong. And in Texas, as all of you know, there was banned abortions. And I think someone else answered in 2011, 42% of unintended pregnancies ended in abortion. So it's a very, very high percentage. And as we all know, physicians know this, unintended pregnancies are actually extremely high risk, right? And so for both the patients and for the baby as well. So I think that there is a huge argument to be made here on why male contraceptives are important for reducing the abortion rate.
There's a question from Orania. You're welcome to ask Orania Contras. Please.
Hola, ¿cómo están? Hello, John. Yes, I'd like to ask if there is or exists a production of antibodies, antisperm, in this patient, like a patient with vasectomy?
Yeah, that's a great question. And it's a question we receive quite frequently as well from urologists and physicians. So the first thing I will tell you that to be completely transparent, we have not measured the anti-sperm antibodies. Again, we've done mostly preclinical research, and that's a little bit harder to do in animal models. However, I don't think that the antibody rate is going to be maybe as high for our product, but also it's as important because really the antibodies and their development really happens long term. That's something that happens over, we're talking 5, 7, 10 plus years. And with our product, we're not really shooting for occlusion for 5, 7, 10 years, at least initially. We're really shooting for occlusion for 1 year or 2 years. And so, I think anti-sperm antibodies, it's an important factor, but and we will measure them in our clinical trials, but I just don't think that long-term they're going to be a huge effect on the reversibility compared to a vasectomy, for example, where a patient gets a vasectomy and 10, 15 years later, they're trying to get it reversed.
Okay, thank you very much.
Well, here's a question from, go on, Doug.
You're really more trying to promote this as temporary contraception than permanent contraception, right?
Exactly.
Okay, so the young man who's 25 years old, who comes into your office, he says, "Doc, I never want to have kids. I don't want to take any chances at all." How do we, when we think that because of his youth, his risk of regret with traditional no scalpel vasectomy could be high given his youth, would we steer him toward Contraline? And I almost have a feeling from my experience trying to steer men toward sperm storage that I might get a little kickback. They can't predict the future, but they're sure they're not going to want to have children.
Yeah, well, so I think the first thing is, we are going through the FDA approval process, and we're not planning on making any claims. And you as a doctor are not going to make any claims that we don't have clinical proof to show, right? So by the time we're on the market, we're going to have an intended use that says, our product Adam is blank percent effective at causing azoospermia for this period of time and has shown to be non-permanent, and it takes this long for the man to regain fertility, right? And that's going to be done in a statistically significant number of men that study. So hopefully for first and foremost, I think that we're going to have a lot of data around this product and help answer some of those questions. I think the other aspect is, again, hydrogels are extremely safe and they're also degradable. They're already used for a number of other applications where they degrade inside the body. My question is not, is this thing going to be permanent or not? It's just going to be, can we make this really effective and then degrade in a very short window where we can say, hey, this is effective for 1 year, and then it takes X number of months to regain fertility, right? And I think that's going to be the bigger challenge.
There was another question, says, what might the risk be of traumatizing the vase while injecting Adam and transforming a non-permanent technique into a permanent one?
Yeah, that's a great question. So I think that, and it's something obviously we're actively studying in our preclinical and eventually clinical trials as well. The only, what we're doing is we're literally putting a 24 gauge catheter into the lumen of the vas deferens. And what we have found is that we have veterinarians who are doing this, that it takes them 1 or 2 tries, and then they're doing it like the back of their hand. It becomes much easier and quicker. And it's something that with little practice, pretty much anyone can do really quickly. And so we've looked at the histopathology of the dogs who have gotten Adam. The histopathology looks very similar, if not better than a vasectomy. You see lumen dilation. You see we have a very low granuloma rate. I think we've only ever seen 1 granuloma. We have not seen hematomas. We haven't seen infection. So the side effect profile in these dogs has been really, really good. And I think that we just the gel itself is very biocompatible, so that's not going to cause any permanent occlusion or fibrosis or anything that we're aware of.
I'm going to give the last question to Chick Wilson. I see you have a question, Chick. Go right ahead and ask. You're on mute, though. You got to unmute yourself.
Unmute.
There
we go. Hey, yeah, thanks, Kevin. Thank you for that presentation. 2 questions. One is I was involved in some of these silastic trials that never succeeded. Why do you think that the hydrogel would be more effective than the silastic, the plugs, the shugs, and those things? Apparently, what's happening there is that sperm just get around them, and they don't obviously stick to the mucosa, and the muscularis of the vas can expand and contract, and what makes you think you're going to be able to block the vas? You can block it. You can't drive a car through it, but if you're a sperm and you got someplace to go, they tend to get out. The other comment was that as we do, tens of thousands of vasectomies, we see these vases that vary in their structure from as small as 1.2 millimeters to over 4 millimeters. And the lumen's always pretty narrow, but cannulating them is not necessarily very easy. Certainly not for the inexperienced. So that and you're trying to inject the same volume into each patient. And my question is, wouldn't we really want to kind of individualize it in each patient? That wouldn't be easy to do from a regimen standpoint.
Yeah. Both of those are great questions. So to start with the latter, I guess, what we have found, as you said, is that the lumen of the vas deferens is actually fairly consistent compared to the outer diameter of the vas deferens, right? And so our goal, at least initially, until something tells us otherwise, is to find out what is the minimum volume that works across all patients, right? And so because and that's kind of what we're trying to figure out, and we haven't answered to that, but I think that is that's what the approach we've taken. I think it would be much harder to regimen this based on the different patients, and I think you're going to get that's where you're going to get varying results.
With that volume,
and-
Kevin, what length of VAS is does that occupy?
Plug, yeah. To be honest with you, it's a little bit confidential, and so at this point, we're not going to disclose that. But you know, you will know at some point. And so, sorry, Chick. This is one of the tough parts about having a company. I can't tell you everything, but in terms of the second question about cannulating, you know, I think that your point is spot on. It's the number one question we get. You know, how successful can a doctor be cannulating the vas deferens? You know, we have found that it's pretty easy with practice. The other aspect is that you could use ultrasound. So it's very easy to use ultrasound and look at the lumen and know you're inside. That's something we've also done. By the way, as a side note, I didn't mention this, but this gel is also ultrasound visible. So you could actually see the gel after implanting it. That's a cool little feature. And the third thing is we're actively making innovations about how can we potentially even automate the delivery, including the needle placement. And so that's something that we're working on, and maybe we can even kind of reduce that risk even further. So I think there's more to come in the coming years for us.
Well, that sounds good. Unlike the results from other types of implants where there's a lot of variability as to how well they were implanted, you could document during your studies with ultrasound that the implant was actually in the right in the lumen. But the first question was actually about how you know this stuff's going to block the vas from sperm.
Oh yeah, and I didn't answer that. My apologies. So I talked about this a little bit, but this gel that we've created, we call it dynamically responsive. So it's extremely durable. I mean, we have models where we plug up, you know, a vas model, and we're trying to inject, you know, 10 pounds, 12 pounds of saline and try to dislodge the gel, and it doesn't go anywhere. So from a durability perspective, it's very, very, very sturdy. The second aspect is that this gel swells. And so that's the beauty of a hydrogel, is that it's actually able to expand with the vas deferens, and it's elastic. So it's able to expand and swell along with it. So that's not the same as silicone or some of those other materials you mentioned. So this gel was made to be expandable is basically my answer to that.
Expandable once or dynamically expandable?
Dynamically. So we have experiments where we can actually, they're called rheology experiments, where we can basically apply a huge amount of torque and friction to the gel and basically almost try to ruin it, and then see how that gel comes back to normal levels, back to normal mechanical properties. And it's pretty remarkable. So this gel is, it's more like tissue than it is this preformed material.
Hey guys, I hate to interrupt because I know we could go on for a long time about this, and I knew it was going to be a popular topic, but we're already at time, and there are a couple of other things we wanted to do before closing out. So please don't everybody leave. Thank you, incredible. Thank you, Kevin, for the amazing presentation. Thank you,